Welcome back to the FARA interview series where we are taking you behind the scenes at the 2019 International Ataxia Research Conference. I am here with Layne Rodden and Dr. Sanjay Bidichandani, and we're here to talk about epigenetic silencing, I believe, and we'll let everyone know what that is. But first of all, we want to do a little introduction. Layne, tell us about yourself.
Sure. I'm Layne Rodden. I'm a PhD student, and I've been working with Dr. Bidichandani for the last 4 years. Our lab is at the University of Oklahoma Medical Center in Oklahoma City, and we only focus on Friedreich Ataxia in our lab, and we're interested in, like Kyle said, epigenetic silencing of the frataxin gene.
So I'm Sanjay Bidichandani. I'm the head of genetics at the University of Oklahoma College of Medicine, and I run a research lab there, and Layne is part of my lab, and we study the epigenetics of Friedreich Ataxia.
Yeah.
All right, so Layne, we heard your presentation this morning. Great, by the way.
Thank you.
Sanjay, we're looking forward to yours tomorrow morning. Both of them deal with this concept, epigenetic silencing. So could you fill in the audience at home about what your presentation was about, Sanjay, what yours is going to be about, and then what epigenetic silencing is in a nutshell?
Yeah, you want me to go first?
Yes.
Yeah. So we all know that people with Friedreich's ataxia have a genetic mutation, okay? And so we understand genetic because what that means is that we have a gene, one we get from mom, one we get from dad, and both of these have a mistake in them, right? So you've got this expansion.
This is the trinucleotide
This is the
repetition.
trinucleotide repeat,
Okay.
the GAA repeat, right? And while that is the primary cause for FA, so if you have this, we know you will have FA. The main question is, now that you have this change, what does that actually do to your gene, right? So we've known for a long time that this GAA expansion shuts the gene off, okay? And we figured out, we and many other people have figured out that the mechanism by which this gene is shut off is what's called an epigenetic mechanism. Now, when we say genetic, you're actually changing the DNA. So you go from GAA 10 times to GAA 1,000 times. But when you're saying an epigenetic change, what you're doing is this 1,000 GAAs just gets wrapped up really tight. So you're not changing the sequence. It's still GAA 1,000, but it just gets wrapped up really tight, and now the cell cannot read it.
And that's like a ball of yarn with a big knot in it.
Exactly. So the DNA is normally just rolled up around these balls, and these balls just get totally wrapped up, and now the cell cannot read the DNA. It's there. You should be able to read it. In fact, all the information needed to make the protein, that's the main function of that gene, all the information is there, but it's so tightly wrapped up that we can't read it.
And is that called a chromatin?
So the whole DNA wrapped around these balls, that's called chromatin, and you can get the U-chromatin, which is kind of the open version, which can be read, or you have this repressive chromatin, which is completely closed up, repressive, it's repressed. And so the point here is, how does that repression actually happen, and how you can untie that to make it become open is the basis of our research.
That's epigenetics in a nutshell.
Exactly.
All right. Layne, tell us a little bit about your presentation.
Okay. So just to branch off from what Dr. B was talking about, we study these epigenetic signals, and initially when we're studying these things, we do them in cells. We have skin cells and we have blood cells from people who have FA. But then the next step after we characterize it in our cell models is to look more in depth in an animal model. So that's what my talk was about today, is looking at these epigenetic signals in different tissues that are important for the pathology of FA, like brain tissue and heart tissue in an animal model.
And you also talked about methylation in a certain region of this gene. Could you tell us a little bit about what methylation is and why that's important to this discussion we're having about epigenetics?
Yes, so DNA methylation is kind of a fancy term for an epigenetic signal that Dr. B was talking about. So cells have several signals and molecules and ways to tell the gene to either be relaxed, like the state that we talked about first, or to be very condensed. And DNA methylation is what we call a silencing signal. So when methylation is placed on the gene, that's one way that the chromatin becomes condensed and inaccessible to the machinery of the cell.
So methylation epigenetics is like an off switch for the gene more.
Exactly.
Exactly.
I saw some of your slides, and we've known for a long time the mechanism of FA is that the more repeats you have, the earlier your onset is, the worse your symptoms are in a general sense. I was interested to see your slides from your research showing that the more methylation you have, that also has a direct relation to some of these other metrics.
That's right. So that's what we found in our cell models, and also the FA animal model that we looked at. The mice that have longer repeats, and also humans who have longer repeats, have more of this epigenetic silencing signal that we call DNA methylation. Okay. So this helps us to learn more about the mechanism of why this repeat expansion is turning the gene off.
So is it less of an off switch and more of a dimmer? The more methylation you
Yes,
have,
actually, yeah.
less it works? Interesting. One other piece, and I don't mean to put you on the spot, I was interested that methylation in heart tissue seems to have less of an expressive impact than methylation in other tissues. Share with us why that might be?
That's right. So that was our big finding from this project, is that it seems like the mechanisms that are silencing the gene in the heart are a little bit different than they are in the other tissues that we looked at, in the brain tissues or in the muscle tissue that we looked at. And we're still trying to figure out what that means for people that have FA, for the cardiac phenotype, for the cardiomyopathy. And so that's what we're trying to look at now, is why is the frataxin gene regulated differently in the heart than it is in the other tissues, and what can we learn from that, and how can we apply that to therapeutic strategies?
In terms of the impact that this would have on the FA community, epigenetics kind of hovers over a lot of these discussions that we're having. Over the discussion we had this morning with Dave Corey and Merrick Naparella on ASOs, over some of these, the Syntef type of molecules, things along those lines. Could you comment on the impact, the scope of epigenetics as it relates to the FA community?
Me?
Either one.
Yeah. I mean, first of all, I think there's no argument about the main mechanism by which the gene is shut off. Okay, it is shut off by an epigenetic mechanism. And so Layne was saying earlier that DNA methylation is just one of these signals that tells the gene that you've got to be off. There are many other signals that are telling the gene to be off. We're just using DNA methylation in this case as a surrogate for that, right? So when you're looking at different tissues over time and seeing if it changes, you've got to pick one of these signals to kind of to just sort of figure out what's actually changing. And so while Layne picked methylation, DNA methylation, others are picking other signals. So our loop is one of those signals, heterochromatin is one of those signals, and so everybody picks their favorite signal to kind of track what's happening. Now, having said that, all of these separate signals actually together constitute what's called epigenetic silencing. So you can treat any of these signals as therapeutic targets, right? So Corey, for example, uses oligos to target the R loop, and one of the reasons why epigenetics causes the gene to be off is it kind of stalls the engine that's supposed to make this gene come on, and Syntef1 is trying to get this engine to move through this epigenetic silencing. So everybody's got sort of different handles on this epigenetic silencing mechanism.
Tell us a little bit about your talk tomorrow morning at the International Ataxia Research Conference.
Yeah, so we've been talking all this time about how the gene is silenced with an epigenetic mechanism, and while Layne was talking to us about DNA methylation as being one of these signals, at the end of the day, when you think about what is actually causing the silencing of the gene, I kind of did this weird thing of saying the whole gene is wrapped up, and actually that's not entirely true. There's a part of the gene that is actually wrapped up like this, and we know what that part is, and we know it based on DNA methylation, based on R loop formation, based on other signals, and so we know exactly where this wrapping up thing is. And what we've discovered, Layne and I, is that region has what's called an enhancer function. So it's the part of the gene that is telling the gene, hey, you got to be on now. You got to be on at this volume, okay? And so the fact that we have this wrapping up or epigenetic silencing at this location,
on this
enhancer
at the master control.
At the control, exactly. And actually, if you scan the whole gene, there's only one control location, and it's here, and this is where that wrapping up is. So that's the basis of my talk tomorrow. And actually, you'll see when people have less epigenetic silencing, they have less wrapping up over here, and you see more function. Okay, and in fact, Layne has figured out how to measure function of that enhancer, and you can see that that function of that enhancer is less in people with FA. So it's actually more of a proof that that is an important regulator of what's going on. So it's exciting to have found sort of the micro location within the gene. So when we are starting to think about therapeutic targets, not only are you targeting the mechanism, but now you're also getting a site location to actually hit the therapy too.
So how we understand now what epigenetic silencing is and maybe how it relates to a bunch of different things, right? So how does this translate into a therapeutic target? Or does it just kind of inform everything? I mean, or is there a specific target that you guys might be working on?
You know, at the simplest level, when you think about it, you know, the gene is off. We got to turn the gene back on. Right? It's a very simple concept if you think about it, right? And if you want to think about the gene being off, really what you're thinking about is where's that dimmer, right? And if you talk to people in the field, there are probably one or two dimmers, okay? And so one or two dimmers means even if you have two dimmers, these are two therapeutic targets to hit, right? So anybody who's studying any of these epigenetic signals or dimmers is basically trying to identify what the therapeutic target is and any clues as to how we might be able to target that actual dimmer signal. And so we've now finally figured out that this is an enhancer sequence. So what that means is it just adds more relevance to this is where a dimmer would be. I mean, this is the signal that's regulating this. That's where, if you had the dimmer somewhere else, you'd have to explain how is that dimmer actually, you know, affecting the gene function. It just happens to be in a place where a dimmer would have a function.
So here the dimmer lines up with the repeat, lines up with the master control. It's all in one nice, tight, inaccessible package.
Yeah.
All right. Sanjay, you've been involved in the FA community for a while. You may not know, but he was on the original paper that described the gene. You're on FARA's scientific board of directors. You run a lab. Layne, you've worked in the FA space your entire lives. Could you describe to the FA community a little bit why FA is so important to you? Why did you choose this disease? Why is it important that patients come to your labs? Things like that.
Should I go first? Yeah. So when I started graduate school, I had to choose a lab out of maybe 50 different labs at the school that I go to, and I personally didn't have a connection with FA before I started grad school. I didn't know anyone personally that had FA. But when I worked in Dr. B's lab for a couple of weeks to decide if this is the place I wanted to do my project, it became very apparent to me early on how involved the FA community was with the research side of things. And this was something that was incredibly unique to Dr. B's lab that I couldn't get from a different lab on that campus. And one of the things that we did was we would have FA families who live in the area come to our lab and see what we're doing, where are the treatments, why are things taking so long. And I really liked the idea of having to think about my research project in a way that I could explain it to people in a meaningful way that are very interested in cures and in treatments and for these FA families. And then another thing that I saw from the FA community very early on was how ambitious everybody involved in the community was. Yeah. And it was just apparent to me that something big and something good was going to happen with this community, and I very much wanted to be a part of that.
Cool.
Yeah.
So, obviously you mentioned that I've been in the FA space for a long time, and I actually, predate the existence of FARA, right? And
it's paleologic at this point.
Exactly. Yeah. I'm 100 years old. And, yeah. So, but I'd finished. Actually, just very quickly. So I'd finished my medical school and graduate school, and I had come to the U.S. for the first time. This is 1994 to do my postdoc fellowship. And that's typically what you do. Once you've finished your medical school or graduate school, you do a postdoc fellowship, and that's how you launch your research career. And typically what you do in your postdoc fellowship determines what you're going to do in the rest of your life. And so when I actually started my postdoc fellowship, it was not in FA, it was in another condition. But when I was in this lab where Massimo was there, Massimo had his team of people who were looking for this FA gene. And to me, that was a very exciting project at the time, just from a scientific standpoint. And so I got involved in that. And of course, I just happened to be in the right place at the right time. And within a year, the gene was found. I contributed obviously to some extent to making that happen. But when that gene was found, it was for the first time a change like this ever was found in any human genetic condition.
Yeah.
Okay, so scientifically it was really exciting, but and you'll hear from Ron Bartek and people like that. It was at that time that they were starting to think about making FARA, right? And what happened is FARA came together and they put together their first grant application, and I actually got the first grant from FARA, the first grant that FARA. So that's how old I am, right? And so I would have to say that being having done my fellowship training in FA and then having received that first grant from FARA, that just cemented my obviously my relationship with FA, but also with FARA. You meet Ron Bartek, you meet Massimo, you meet these guys. Dave Lynch, you're never going to leave FA, right?
Yeah,
and
absolutely.
frankly, after that, I've met many families, and I'll also say one more thing, FA is a very unique condition in that it usually affects kids, but not very young kids, and so you're affecting people who are able to communicate directly with you what it is they have, right? So if you're dealing with a muscular dystrophy, usually you're speaking to the parents. If you're speaking to adults who have an adult onset condition, you're speaking to a spouse or someone who's lived through life and has seen many things. But here you're dealing with young teens or young people who are grappling with life in general, all the challenges that life throws at them. But at the same time, FA is one of those things. And that is actually especially motivating and energizing to kind of just be part of that community and part of that feeling. So I hope that some of the things that Layne was saying has obviously been transmitted to the next generation, and so hopefully we've infected enough people with the FARA virus.
Well, you guys made me have a little bit of a Ron Bartek moment. Layne made me tear up a little bit. So good on you. Thanks for doing that. Thanks for putting your heart into what you're doing, and we really appreciate your work.
Yeah,
Thank you.
We're so lucky and blessed to have you guys and have so many other amazing researchers and folks involved in this fight to stop this ridiculous disease.
Yes,
We're
absolutely.
happy to be a part of it.
Yeah, well, we're happy.
We'll get there.
We will.
Yeah, we'll
We
get
will.
there. Yeah.
As has been said, we can get this one. So thank you all for continuing to tune in. Be sure to tune in next time when we interview Dr. Mark Payne and Michael Clark on Tat for Taxin, a topic that's of great interest to the entire community. Check us out on FARA's Facebook page, on Twitter, on LinkedIn, and on the YouTube channel. Until next time, we'll see you.
Bye-bye.
Yep, no problem. Take two.
Oh. There's a technical reason for that.